Hello and welcome dear students. Today we are going to discuss the topic on disorders associated with defects in protein, amino acid metabolism and with deficiency of urea cycle enzymes. The objectives of the present study are to study the metabolic disorders Phenylketonuria, Alkaptonuria, maple syrup urine disease, Homocystinuria, Tyrosinemia, urea cycle and urea cycle disorders. Now, let us start with a brief introduction. Metabolic diseases are any of the diseases or disorders that disrupt normal metabolism, the process of converting food to energy on a cellular level. This process is carried out by thousands of enzymes participating in numerous interdependent metabolic pathways. Metabolic diseases affect the ability of the cell to perform critical biochemical reactions that involve the processing or transport of proteins, carbohydrates or lipids. One group of these disorders is amino acid metabolism disorders. Amino acids are the building blocks of proteins and have many pivotal functions in the body. 20 amino acids, including 9 that cannot be synthesized in humans and must be obtained through food, are involved in metabolism. Amino acids function as are synthesized into important molecules in the body such as neurotransmitters, hormones, pigments and oxygen carrying molecules. Each amino acid is further broken down into ammonia, carbon dioxide and water. Hereditary disorders of amino acid processing can result from defects either in the breakdown of amino acids or in the body's ability to get amino acids into cells. The disorders related to the metabolism of amino acids include Phenylketonuria, Tyrosinemia, Homocystinuria and maple syrup urine disease. These disorders are autosomal recessive and all can be diagnosed by analyzing amino acid concentrations in body fluids. Now, Phenylketonuria. Phenylketonuria occurs in infants born without the ability to normally break down an amino acid called phenylalanine. Phenylketonuria is caused by lack of the enzyme phenylalanine hydroxylase needed to convert phenylalanine to tyrosine. Phenylketonuria is a disorder that causes a build-up of the amino acid phenylalanine, which is an essential amino acid that cannot be synthesized in the body but is present in food. Excess phenylalanine is normally converted to tyrosine and eliminated from the body. Without the enzyme that is required to convert it into tyrosine, phenylalanine builds up in the blood and is toxic to the brain, causing intellectual disability. Now, what are its symptoms? Due to a decreased amount of the pigment melanin, persons with phenylketonuria tend to have lighter features such as blonde hair and blue eyes than other family members who do not have the disease. Other symptoms include seizures, nausea and vomiting, an eczema-like rash, aggressive or self-injurious behavior, hyperactivity and sometimes psychiatric symptoms. Now, next is Alkaptonuria. Alkaptonuria is a rare autosomal recessive inherited disorder originally defined as an inborn error of metabolism by Archibald Garrod in 1902. Alkaptonuria is caused by defects in the gene encoding an enzyme homogenistic acid oxidase involved in the catabolism of phenylalanine and tyrosine. As a consequence of this gene defect, the catabolism of these two amino acids is inhibited and the intermediate homogenistic acid is excreted in the urine. A buildup of homogenistic acid causes the urine to turn dark blue or black when exposed to air as a result of the conversion of homogenistic acid to a melanin-like compound. Alkaptonuria is characterized by homogenistic aciduria, ochronosis, that is a bluish-black discoloration of tissues, and arthritis. Now, what are its causes? Alkaptonuria is caused by a mutation on homogenetic 1,2-dioxygenase gene. The homogenistic acid oxidase gene is found on chromosome 3q13.33 spanning 60 kb and encompassing 16 exons that encode a protein of 445 amino acids. A number of single nucleotide changes, insertions, deletions and mutations in introns have been identified in alkaptonuric patients. In all, mutations have been found in 11 of the 16 exons of the HDG gene. Also, it's an autosomally recessive condition. This means that both of your parents must have the gene in order to pass the condition on to offspring. Now, coming to its symptoms. The three major features of Alkaptonuria are the presence of HGA in the urine, ochronosis, and arthritis of the spine and large joints. Dark stains on a baby's diaper are one of the earliest signs of Alkaptonuria. Symptoms become more obvious with age. Alkaptonuria can also lead to heart problems. The build-up of homogenistic acid causes heart valves to harden. This can keep them from closing properly, resulting in aortic and mitral valve disorders. In severe cases, heart valve replacement may be necessary. The build-up also causes blood vessels to harden. This raises the risk of high blood pressure. Now, next is maple syrup urine disease. Maple syrup urine disease is a metabolic disorder caused by a deficiency of the branched chain alpha keto acid dehydrogenase complex, leading to a build-up of the branched chain amino acids like leucine, isoleucine and valine and their toxic byproducts, that is keto acids in the blood and urine. In this disorder, the body is not able to break down the amino acids leucine, isoleucine and valine. These amino acids are found in protein-rich foods like meat, fish and eggs. Byproducts of these amino acids build up, causing neurological changes, including seizures and intellectual disability. These byproducts are the reason that causes body fluids such as urine and sweat to smell like maple syrup. This disease is most common among Mennonite families. Now, next is homocystinuria. Homocystinuria is caused by a defect in cystathionine beta synthase, an enzyme that participates in the metabolism of methionine, which leads to an accumulation of homocysteine. This disorder can cause a number of symptoms, including impaired vision and skeletal abnormalities. Children with homocystinuria are unable to metabolize the amino acid homocysteine. Infants with this disorder are normal at birth. The early symptoms, including dislocation of the lens of the eye, causing severely decreased vision, usually begin after 3 years of age. Most children have skeletal abnormalities, including osteoporosis. Children are usually tall and thin with a curved spine, chest deformities, elongated limbs and long spider-like fingers. Homocystinuria makes the blood more likely to clot quickly, resulting in strokes, high blood pressure and many other serious problems. Next is Tyrosinemia. Tyrosinemia is caused by lack of the enzyme needed to metabolize tyrosine. The most common form of this disorder mostly affects the liver and the kidneys. Children with tyrosinemia are unable to completely metabolize the amino acid tyrosine. There are 3 main types of tyrosinemia: type 1st, type 2nd and type 3rd, each with deficiency of different type of enzyme and symptoms. Tyrosinemia type 1st is most common among children of French Canadian. Classic tyrosinemia is caused by a deficiency of fumaryl acetoacetate hydrolase, the last enzyme in tyrosine catabolism. Features of classic tyrosinemia include severe liver disease, unsatisfactory weight gain, peripheral nerve disease, and kidney defects. Approximately 40% of persons with this disorder develop liver cancer by the age of 5 if untreated. Treatment with 2-nitro-4-trifluoromethylbenzoyl 1,3-cyclohexanedione, a potent inhibitor of the tyrosine catabolic pathway, prevents the production of toxic metabolites. Tyrosinemia type 2 is caused by a defect in tyrosine amino transferase leading to the plasma tyrosine concentration typically greater than 500 micromole per liter that may exceed 1000 micromole per liter. Affected individuals have painful and hyperkeratotic plaques on the soles and palms. Type 2 tyrosinemia is less common. Tyrosinemia type 3, the rarest of the tyrosine disorders, is caused by a deficiency of p-hydroxylphenylpyruvic acid dioxygenase. Plasma concentration of tyrosine ranges from 350 to 650 micromole per liter. It is characterized by the increased excretion of 4-hydroxylphenylpyruvic acid, 4-hydroxyphenylactate, and 4-hydroxyphenylacetate. These individuals, like those with tyrosinemia type 2, have no liver involvement but have skin or ocular changes. Now, coming to the urea cycle. The urea cycle involves a series of biochemical steps in which nitrogen, a waste product of the protein metabolism, is removed from the blood and converted to a compound called urea in the blood. Urea cycle involves 5 catalytic enzymes. First, carbamoyl phosphate synthetase. Second, ornithine transcarbamylase. Third, organosuccinic acid synthetase. Fourth, organosuccinic acid lyase. And fifth, arginase. A cofactor producing enzyme, N-acetyl glutamate synthetase. Urea cycle disorders result from inherited deficiencies in the 6 enzymes of the urea cycle pathway. The urea cycle also depends on 2 transporters, ornithine translocase and citrine. Abnormalities in these transporters can lead to hyperammonemia. The liver is the only site of the complete urea cycle. Among the 6 enzymes in the cycle, N-acetyl glutamate synthase, carbamyl phosphate synthetase first, and ornithine transcarbamylase are intramitochondrial, whereas organosuccinate synthetase and organosuccinate lyase are cytosolic. Now coming to urea cycle disorders. The urea cycle disorders result from defects in the metabolism of waste nitrogen from the breakdown of protein and other nitrogen containing molecules. Severe deficiency or total absence of activity of any of the first 4 enzymes in the urea cycle or the cofactor producer results in the accumulation of ammonia and other precursor metabolites during the first few days of life. Infants with a severe urea cycle disorder are normal at birth but rapidly develop cerebral edema and the related signs of lethargy, anorexia, hyper or hypoventilation, hypothermia, seizures, neurological posturing, and coma. In milder deficiencies of these enzymes and in arginase deficiency, ammonia accumulation may be triggered by illness or stress at almost any time of life. These diseases include arginino-succinic aciduria, arginase deficiency, carbamyl phosphate synthetase deficiency, citrullinemia, N-acetyl glutamate synthetase deficiency, ornithine transcarbamylase deficiency. Now coming to organosuccinate lyase deficiency. Organosuccinate lyase deficiency affects the body's ability to clear the nitrogen that is incorporated into the urea cycle as organosuccinate. This causes hyperammonemia. Severe defects often represent rapid onset hyperammonemia in the newborn period. This enzyme defect is past the point in the metabolic pathway at which all the waste nitrogen has been incorporated into the cycle as arginine. Treatment of these patients is based on a reduction in nitrogen intake and supplementation with arginine to complete a partial cycle. This disorder is marked by chronic hepatic enlargement and elevation of transaminases. Biopsy of the liver shows enlarged hepatocytes which may progress over time to fibrosis. Deficiency of this enzyme prevents the conversion of succinate to the amino acid arginine, which affects the urea cycle and other biochemical pathways. Untreated babies with ASA often die within the first few weeks of life. Early treatment can help prevent serious problems including brain damage, liver problems, and intellectual and developmental disabilities. These are problems with how the brain works that can cause a person to have trouble or delays in physical development, learning, communicating, taking care of himself, or getting along with others. Now, coming to arginase deficiency. Arginase deficiency is not typically characterized by rapid onset hyperammonemia. These patients often present with the development of progressive spasticity with greater severity in the lower limbs. They also develop seizures and gradually lose intellectual attainments. Growth is usually slow, and without therapy, they usually do not reach normal adult height. Diagnosis is made by the elevated levels of arginine in the blood and by analysis of enzymatic activity in red blood cells. Treatment is similar to other urea cycle disorders. Limitation of protein, use of essential amino acid supplements, diversion of ammonia from urea cycle, with the exception that arginine supplementation is not indicated due to the excessive arginine levels already present in this condition. Now coming to ornithine translocase deficiency, that is triple H syndrome. The hyperornithemia, hyperammonemia, Homocystinuria syndrome is an autosomal recessive inherited disorder. Plasma ornithine concentrations are extremely high. The defect in ornithine translocase results in diminished ornithine transport into the mitochondria with ornithine accumulation in the cytoplasm and reduced intramitochondrial ornithine causing impaired ureagenesis. Growth is abnormal and intellectual development is affected. Now, next is N-acetylglutamate synthetase deficiency. N-acetylglutamate synthetase deficiency affects the body's ability to make N-acetylglutamate, which is a required cofactor for the function of carbamyl phosphate synthetase first. Without N-acetylglutamate, CPSC1 cannot convert ammonia into carbamyl phosphate. Along with OTC deficiency and CPS1, deficiency of N-acetylglutamate is the most severe of the urea cycle disorders. Patients with complete N-acetylglutamate synthetase deficiency rapidly develop hyperammonemia in the newborn period. Next is carbamyl phosphate synthetase first deficiency. Carbamyl phosphate synthetase first deficiency affects the liver's ability to convert nitrogen to urea. This enzyme takes ammonia and through the use of bicarbonate and ATP produces carbamyl phosphate. This enzyme requires the presence of its cofactor N-acetylglutamate. Along with OTC deficiency and N-acetylglutamate synthase, deficiency of CPS1 is the most severe of the urea cycle disorders. Patients with complete CPS1 deficiency rapidly develop hyperammonemia in the newborn period. CPS1 is the first enzyme in the urea cycle and is found primarily in the liver. It requires the cofactor N-acetylglutamate to function. Currently, diagnosis is based on enzymatic assay of liver tissue. Sequence analysis is available on a research basis only. Next is succinate synthetase deficiency or citrullinemia first. Defects in succinate synthetase affects the ability to incorporate ammonia into urea. This enzyme combines citrulline with aspartate to form succinate. Patients with complete ASSD present with severe hyperammonemia in the newborn period. The use of arginine in these patients allows some nitrogen to be incorporated into the urea cycle, which makes treatment somewhat easier than other defects in the cycle. Citrulline levels in these patients can be hundreds of times the normal values. Unlike CPS1, NAGS and OTC, this enzyme is distributed throughout the body. Diagnosis is by enzymatic assay of fibroblasts. Next is citrine deficiency or citrullinemia second. Citrullinemia second is an autosomal disorder that results in decreased activity in the liver of a transport molecule for aspartate. This results in limitation of activity for the enzyme argininosuccinic acid synthase, which combines aspartate and citrulline to make argininosuccinic acid. Citrine is an aspartate glutamate transporter across the mitochondrial membrane. This defect can present with classic newborn hyperammonemia, jaundice and fatty liver, but is more likely to present with insidious neurologic findings, hyperammonemia, hypercitrullinemia and hyperlipidemia in adulthood. Now, next is ornithine transcarbamylase deficiency. Ornithine transcarbamylase deficiency affects the liver's ability to convert ammonia into urea. Along with CPS1 and NAGS deficiency, OTC deficiency is the most severe of the urea cycle disorders. Patients with complete OTC deficiency rapidly develop hyperammonemia in the newborn period. Patients with partial OTC deficiency can present at almost any time of life with a stressful triggering event. Diagnosis is based on the presence of large amounts of argininosuccinic acid in the bloodstream and direct enzymatic analysis of fibroblasts. Now coming to neurologic aspects of urea cycle disorders. Ammonia can cause brain damage through a variety of proposed mechanisms, a major component of which is cerebral edema. The specific roles of ammonia, glutamate and glutamine in cerebral edema are still under investigation. They are thought to affect the aquaporin system and water and potassium homeostasis in brain. Chronic hyperammonemia may disrupt ion gradients and neurotransmitters transport of metabolites, mitochondrial function and the alpha ketoglutarate or glutamate/glutamine ratio. Seizures are common in acute hyperammonemia and may result from cerebral damage. Recent findings suggest that subclinical seizures are common in acute hyperammonemic episodes and their effects on cerebral metabolism. With this, we come to the end of today's lecture. Thanks for watching.